Our next talk is by Dr. Mark Anderson, who comes to us from UC San Francisco. And he is going to talk about what type 1 diabetes research can learn from APS type 1. Thanks, Mark.
Thank you, Jennifer. It's always a pleasure to be amongst the families we're trying to help in the lab, and really a privilege. So in thinking about the meeting, I've been involved in helping plan the meeting, and Jennifer twisted my arm to give a talk about the connection of type 1 diabetes with APS1, and so that's the goal. Before I start going into the details, though, I just wanted to reflect on both the movie and the Wordle that we just saw right now, and I saw that one of the prominent features there was life without doctors, and I'm certainly committed to putting myself out of business. I have no problem with that. And the second thing I say is that I turned 50, I'm 52 now, but when I turned 50, my brother made me one of these Wordles, and I will tell you that AIR and APS1 were in large letters on my Wordle, just so you know, it's definitely part of my personal life and my everyday thoughts. So what I want to tell you a little bit about right now, if you don't want to listen to anything else I have to say, you know, there is this issue about APS1. Yes, it's a rare disorder, and there's this feeling of trying to get organized and advocate for this small rare disease. But the one message I want to give all of you is that you're not alone, right? There are many other diseases that have a connection to AIR, and certainly amongst them is type 1 diabetes. And I think one of the goals of this foundation, and I'm happy to help with facilitating this, is to get the other diseases to recognize the value of working together with the APS1 Foundation and studying what's going on with people with this disease, because they certainly have been incredibly instructive in how the immune system operates. Okay. So for me, I just wanted to start with a clinical case of the very first patient I ever heard of that had APS1. And so this is almost 20 years ago when I was in Boston at Mass General Hospital. And at that time, this woman was 51 years old, and she sort of had the classic features of many things that we've heard about before. So at the age of 7, she developed hypoparathyroidism, got the candidiasis, got the Addison's disease, and this was long before the AIR gene had been found, but because she had those things, the doctors recognized that she had APS1. And then later in her life, she went on to have ovarian failure. She lost her body hair, but then at the age of 40, she developed type 1 diabetes. And it was convincing that it was autoimmune type 1 diabetes because she had the classic autoantibodies that we see in the disease, and she was dependent on insulin. Now, for me, I'm an adult endocrinologist, and I have a PhD in immunology, and this whole thing captured my attention that it's not an accident that this disease is popping up amongst these constellation of symptoms. So to back up for a second, since I was asked to speak about type 1 diabetes, I just wanted to give a little bit of a background about type 1 diabetes. So it goes by other names. So it's also called insulin-dependent diabetes, juvenile diabetes, which is a complete misnomer because there's certainly, just as you saw here, a 40-year-old developing type 1 diabetes. That happens, right? We do get adults that get type 1 diabetes. You'll see a picture in a minute here, but prior to the discovery and purification of insulin in the 1920s, this was a fatal disease. And then the typical presentation of someone that has type 1 diabetes is someone that has frequent urination, extreme thirst, weight loss, and then they can get this metabolic disorder called ketosis. So here's a picture of a child that was fortunate enough to develop type 1 diabetes around the time of the discovery of insulin. And so before insulin had been discovered, the person that developed type 1 diabetes, because they were not making insulin, the way the disorder was described was excess in the setting of starvation, right? They could not use the glucose that their body was absorbing because insulin gives your body cells the instruction to use the glucose. So even though this patient here has type 1 diabetes, has very high glucose levels, the cells in this person's body could not use the insulin. And so, the glucose. And so because they can't use the glucose, they start burning their own protein and fat. And that leads to this terrible wasting disorder. I mean, look at this person. I mean, it looks like somebody that's just basically dying of starvation. And so this particular child, though, developed disease around the time of the discovery of insulin, and you can look at this dramatic effect that insulin had on this person. So besides the fact that the blood sugar gets corrected, it also allows the body to properly use glucose as a substrate for energy and the energy that we need for all our cells to work. Now, I thought that it was also appropriate to just throw in a slide here about the discovery of insulin. So the Orange family, who's played an incredible role in this foundation, are from Toronto. And many of you may not know this, but Toronto's gift to the biomedical world is the discovery of insulin. And so we credit the discovery to these two gentlemen, but it was certainly a team that was at the University of Toronto. So this is Fred Banting and Charles Best. And they were working with the hypothesis that they knew there was some factor that was made in the pancreas that made the hormone that controlled blood glucose. And so by working with dogs, where they would take out the pancreas of dogs, they would then try to replace a purified protein into the dogs that would control the glucose. And that's ultimately what led to the discovery of insulin. But another interesting thing about the story I'll just throw in is in 1922 was the first time that insulin was ever injected into a human being, and that was done in Toronto. But look at this, within a year, commercial insulin was manufactured and distributed through the world. And in 1923, the Nobel Prize was awarded to Banting and the lab boss of these two gentlemen, MacLeod, which also has its own unique story to it. All right, so just real quickly about how type 1 diabetes works and how diabetes works in general. This is just a slide about how glucose metabolism normally works in our body. So when we are in our resting state, when we're not eating, we have the pancreatic islet here, which is greatly enlarged, right? And where glucose is coming from in the body. So our liver makes glucose all the time. And glucose is an important substrate for our cells to work. Where it's really, really important is actually in the brain. So the brain's main energy source is glucose. And some of this starts making sense. So one of the things you'll hear about with people that have diabetes is if they take too much insulin and they make their glucose level go too low, one of the things that can happen when they have what we call hypoglycemia is they can have a seizure. And the reason why is that there's no energy getting to the brain. The brain has no other way of getting energy. And so besides the brain, the other major places that glucose gets used are in the liver, the muscle, and the fat. So what happens when we eat a meal? So when we eat a meal, obviously a large amount of glucose is coming into our body through the food that we're eating that's getting degraded and chewed up. And then when that happens, signals get sent to the pancreatic islets, including the elevated glucose level, to tell the islets, hey, it's time to make insulin. And when you make insulin, the big thing that it does is it tells the liver, muscle, and fat to use that glucose. All right? So then let's go here to the condition of type 1 diabetes. So what's going on in type 1 diabetes is there's no insulin being made by the pancreatic islet. You got glucose coming in from the liver. There's a counter-regulatory hormone called glucagon, which makes things even worse. But you have all this glucose floating around in your body, but you can't use it, right? The liver, muscle, and fat are not getting the signal to use it. And so that's going back to that patient that you saw back in the 20s of what was going on in her body was that she had all this glucose in her blood, but she's not able to use it. And then ultimately what happens is this starts showing up in the urine and you pee it out. All right, so then going back to type 1 diabetes and sort of the history of type 1 diabetes, why the insulin deficiency? And so when people started looking at patients that died from this disorder and they looked in the islets, they saw that their islets were often atrophied and not there. But it took quite some time to start appreciating that this was actually a problem with the immune system. And so this is just an example of looking at a pancreatic islet in a human being that had been biopsied with someone with type 1 diabetes. And when they went and looked in that pancreatic islet where the insulin is getting made, people were able to detect T cells being present. So this green color is a stain that's specific for T cells. And so you can see these T cells that are specifically infiltrating the islet of this new onset type 1 diabetic patient, right? And so this kind of work that happened initially in the 60s and the 70s came around for us to understand that type 1 diabetes was actually an autoimmune disease, that the immune system was actually actively killing the islets. And so this is just a beautiful diagram of a model of how we think type 1 diabetes occurs in an individual that has it. And so this was developed by my late colleague George Eisenbarth at the University of Colorado. So the way to think about this in an individual, so the beta cells are are the cells that make insulin. So this is the amount of insulin-producing cells that you have in your body. And then people that get type 1 diabetes outside of APS1, they still have genetic predisposition. I'll go into that in a minute. But then we think there's some sort of trigger out there that happens. And this trigger starts activating the immune system to attack the islets in the pancreas, right? And so some sort of trigger activates T cells to start attacking the islets. And as time marches on, one of the first things that we can detect in the blood of people, even before they get type 1 diabetes, is they'll have autoantibodies. And we've heard a lot about autoantibodies already in APS1. And the same thing happens in APS1-mediated diseases. And that is that you can detect the antibodies before the patient is actually getting complete destruction of the organ. And so this is a great model for any of the other autoimmune diseases that you see in APS1. And so these antibodies are probably a great marker of what the T cells are specific for. We're just much better at being able to measure these than we are the T cells in someone's blood. So then as time marches on, you can imagine the immune system is attacking these insulin-producing cells. They're starting to decay away. And then once you attack enough of the insulin-producing cells, patients start having problems with their metabolism of glucose. And so they develop this intermediate syndrome we call glucose intolerance. And the only way that we can detect that is we have somebody eat a meal or take in some glucose, and then we measure their glucose after they do this. And instead of being normal, there's some slight abnormalities into it, but they don't have full-blown diabetes. And then as time marches on, they come to clinical attention. So the important point to make here is that when kids that develop type 1 diabetes come to the clinic and they're peeing frequently, they're drinking water frequently, they may even have the ketosis, this disease has been going on for quite some time before they come to clinical attention. Okay, so going back to type 1 diabetes and the connections to APS1, I just wanted again to give you the message that type 1 diabetes also has a strong genetic component to it. And that's a little bit confusing because frequently when we see a patient in clinic that has type 1 diabetes and we ask about a family history, the most common thing that we hear is that there's no one else in the family that has type 1 diabetes. But with studying this disease over time and looking at it in much greater numbers of patients, there's some simple facts. So actually the best place to start is at the bottom. So type 1 diabetes in the United States affects roughly over 1 million individuals. So obviously there's a much richer resource of patients to look at. But within that 1 million individuals, we've been able to do identical twin studies, right? So we have two people that are genetically identical, and one of them develops type 1 diabetes. What we do know is the lifetime risk of that other identical twin getting type 1 diabetes is around 50%, right? So right there you can see the genes are playing an important role, but they're not everything. Now, if you have a sibling that has type 1 diabetes, your risk of getting type 1 diabetes is about 3 to 5%. And then in the general population in the United States, the risk is about 0.3%. And so the sibling risk is also a great marker of genetic risk. So the sibling risk is about 15 times greater than if you didn't have a sibling with type 1 diabetes, which just again emphasizes the genes are playing an important role. So what's been done? And so over the last 20 years, all this great technology that we've developed with studying the human genome has been applied to this disease, where we've basically taken large collections of people that don't have type 1 diabetes, people that do have type 1 diabetes, and ask what genes are influencing the development of this disease. And so this is a slide that's now more than a decade old, but this is just highlighting some of the genes that we know play a role in the development of type 1 diabetes. And so by far and away, the greatest risk maps to this gene we call the HLA, right? And the HLA, very complicated gene, even for doctors, this is a complicated gene to understand. It has this terrible nomenclature associated with it. But the big point about the HLA is these are the genes that control how T cells see the world, right? So when a T cell sees the world, it sees it on the surface of another cell we call an antigen-presenting cell. And this HLA gene basically controls the expression of those genes that T cells are seeing. And so what we think this means is that certain HLA types put you at much greater risk of developing type 1 diabetes. And it points to the idea that T cells are important in type 1 diabetes. Now what's also interesting though is this second gene right here, right? So the second highest risk gene for type 1 diabetes maps to the insulin gene itself. right? And so the variant in the insulin gene that it maps to is actually not in the coding sequence of the insulin gene, but in the part of the insulin gene that controls its expression. And I'm going to show you a slide about that in a second. And it actually was somewhat of a surprise. The last point I want to make about this slide is there are all these busy names here, but the one thing that I'll tell you is about all these other busy names is the other genes that map to type 1 diabetes are genes that have something to do with the immune system and the control of the immune system. So it's been incredibly revealing in why type 1 diabetes happens is that it's an immune system problem in general. The genes that show up on this list for the most part are not genes that are made in the pancreatic beta cells where insulin is produced. They're perfectly fine. Okay. So now I'm coming back to this variant in the insulin gene, the second highest risk gene in type 1 diabetes, and we give it this very fancy name called IDDM2, which just stands for insulin-dependent diabetes gene risk. And basically the way to think about this is in the human population, the gene comes in two ice cream flavors, vanilla and chocolate, right? And so the vanilla form we call class 1, and the chocolate form we call class 3. And they're in the part of the insulin gene that doesn't code the gene but controls how it gets expressed. And it was somewhat of a mystery as to why this change in how the gene gets expressed relates to risk. Because when people started studying it and looked at the two forms, it looked like that when you looked in the pancreas, there really was no change in how the gene was getting expressed in the pancreas. But in the late 90s, several groups made this really interesting observation, and that was that the gene, or this variant in the gene, controls how insulin gets expressed in the thymus, right? And so this is just, at that time, some technology to look at the expression that came off of one version of the insulin gene versus the other version. So this is an individual that had one copy on one chromosome of the vanilla and one copy on the other chromosome of the chocolate. And what you could see is that the expression of the vanilla version was lower than the chocolate version, right? And so what we came to appreciate is what this risk gene was actually doing was correlating with the expression of insulin in the thymus. And at that time when this was discovered, many people really weren't sure about the role of insulin getting expressed in the thymus. People could say, well, we can find some insulin in the thymus, but we're not sure is that actually important or not. And so one of the simple questions, of course, is how does this thing get expressed in the thymus in the first place, right? The thymus is not a place where metabolism is controlled, unlike the pancreatic islets. And does it have a completely different mechanism? And so along comes air, right, in a patient with the air disease. And this connection of someone that has type 1 diabetes and a mutation in the air gene. And so, as you've heard over and over again, we've come, over the last decade and a half, we really have come around to understand what air is doing in the body. And so the main thing that air is doing is it's expressed in these specialized cells in the thymus. And so this is yet another picture of the thymus where we sectioned it. In the brown color are the cells that are actually making the air gene, the gene that's defective in APS1, right? And it turns out that when we started getting this result, we hypothesized that these are the exact same cells that are making insulin in the thymus, right? And in parallel, not our group, but another group at UCSF, Doug Hanahan had described these rare cells in the thymus that could make insulin. And again, nobody was sure exactly how this all worked. All right. And I apologize for the redundancy. We've been through this already, but I just want to make sure that we're all on the same bandwagon here. So I have my version of the thymus, and it's a school, right? Bone marrow precursors go in there, and out come T cells. And so that's an important job of the thymus, is that it promotes the development of T cells. And then this cartoon, the one thing I just want you to focus on right here is just imagine all these cells with different colors on their surface. Right? And the reason that you have all these different colors is each individual T cell has its own receptor. And the way that that gets made is through a slot machine process where the DNA gets chewed up, spit out, and repasted together. And so the way to think about this, again, are T cells get generated by pulling the slot machine lever. Right? Each T cell has its own receptor. And so that's fantastic, right? That allows you to have all these different kinds of T cells that can go and see the world. But the downside of this, of course, is some of those T cells are going to be reactive against self. And so that comes back to emphasize the importance of the school. So the school is not only important in nurturing and bringing along T cell development, but it also has to tell T cells the difference between self and non-self. And it turns out that that place where that's occurring is in that compartment where air is expressed and where also insulin's expressed. A simple question, of course, came along. Does air play a role in getting insulin made in the thymus? How the heck could we show that that's true, right? And so a number of years ago, when I was a postdoc at Harvard, we made a mouse model of a mouse that's deficient in air, back when it was difficult to manipulate the genome. So we have a mouse that's deficient in air. Basically, this is an APS1 mouse, right? And then when we do experiments, we always have controls, right? So this is a mouse that's otherwise genetically identical and has a normal air gene. And then what we did is we took the thymus out of these mice, and then we chopped up the thymus into a cell suspension where we could purify those cells that are making air from either the wild-type mouse or the knockout mouse, and then we could look at gene expression, right? And then at that time, another amazing thing had come along, and that was our ability to go and measure every single gene, right? At the time, most people had to use a short list of genes just because of the technology, and so this is a technology that's almost become obsolete for reasons I don't want to get into, but this is a picture of a gene chip where we could take the RNA that encodes all the genes and put it on this chip and ask, what are all the genes that are different between these cells and these cells down here, right? And so after doing this experiment, we get this very busy list, and I have no intention of showing you all the details of this list, but these are the top 30 genes that are different between the air wild-type thymic epithelial cells and the air knockout thymic epithelial cells. And then it's impossible for you to see, but where I put this green box on the list of the top list of genes that get regulated is insulin itself, right? And this experiment has been repeated by many, many other groups. So at least in mice, I can say without a doubt, unequivocally, that insulin gets expressed in the thymus, and the major regulator of how insulin gets expressed in the thymus is the air gene. Okay. So one other quick thing I wanted to tell you, of course, is that patients with APS1 have type 1 diabetes autoantibodies. And this is just an example of data where we took or I should say that this is Allah's paper, where he took a large collection of patients with APS1, and then he put it on this fancy array where we can measure all the different autoantibodies that are present. And the main thing I want to point out is over here is are these two antigens called GAD1 and GAD2, and they're associated with reactivity against the islets. And so the main thing to show is many APS1 patients have autoantibodies against the pancreatic islets. And so at some level, this is starting to make sense. Air controls insulin's expression in the thymus. It probably controls the expression of other pancreatic islet antigens in the thymus. And this is probably why there's some prolactivity to not only making autoantibodies against the pancreatic islets, but also why some patients get type 1 diabetes. Now, the doctors like to argue about type 1 diabetes and its prevalence in APS1. I'd say in my estimate, it's probably somewhere around 10% of patients that have APS1 have type 1 diabetes. Now, that may seem low to you, but again, in the general population, the prevalence of type 1 diabetes is less than 0.5%, right? So it is definitely higher in individuals that have APS1. Don't let somebody tell you that APS1, there's not much type 1 diabetes. Now, it is interesting that the prolactivity of making autoantibodies against the islets is much higher. It's more than 50% of APS1 patients make autoantibodies against islet antigens. And so what I wanted to put up here is that this APS1 is actually a rich resource for understanding how type 1 diabetes works. And that's because it's a much more severe disease. It's a much higher prolactivity against the pancreatic islets that's happening in these patients. And so it leads you to ask some other questions. So a simple question is, well, if the incidence is only 10%, maybe there's something going on in these patients that protects them against getting type 1 diabetes. And one paper that was published more than a decade ago was that it is known that one of the genes that protects against developing type 1 diabetes is that HLA gene, that highest risk gene that I described to you earlier, right? So if you have a certain version of the HLA, it actually protects you against getting type 1 diabetes, and that's been observed in APS1. And certainly now with the rapid advance in genetics, we probably have the potential, if we study enough patients with APS1, we could find other genes that might protect against type 1 diabetes. At the same time, we might find genes that actually predispose to type 1 diabetes on top of APS1. Another thing that's controversial right now, or provocative, I would say, are there factors that APS1 patients have that protect them against type 1 diabetes. And so there's some data now to suggest that perhaps antibodies against things like IL-17 and IL-22 could protect people against getting type 1 diabetes. And we're still sorting that out right now. But again, that gives you an example of what could be studied with patients with APS1. So putting this all together, this is a very fancy cartoon from a review that Jeff Bluestone and I wrote a number of years ago about how we think type 1 diabetes happens. And so in this fancy cartoon, imagine this is our crude drawing of the pancreas, and these little circles are the pancreatic islets where insulin gets produced. And then in this cartoon, we're just highlighting how we think the autoimmune disease gets started and where it comes from. And there's way too many arrows here, but I think the main thing to portray is it is a complicated disease, but we're starting to figure out all the pieces. And so what's shown here is that ultimately what happens is you get a pool of these green T cells that get activated that are specific for antigens in the islets, and they kind of direct this destruction of the pancreatic islets. So the big question is, well, where the heck did these T cells come from in the first place, right? And so we know that all T cells come from one place. They come from the thymus, right? And we know that what comes out of the thymus in patients with type 1 diabetes ultimately are T cells that are specific for things like insulin, right? And then through a series of mechanisms, somehow the triggering happens and this thing is let loose. And once it gets going, it's hard to unwind it. But then take the whole picture together, right? We now know that the genetics of type 1 diabetes support that insulin is a major risk gene for the development of the disease. We now know that that risk gene actually is controlling how insulin gets made in the thymus. And then in the last 15 years, we've come around to figure out that there's an intersection with air, right? That patients with APS1 or with air allowed us to make the insight that air is actually made in the thymus under the control of air, right? And this starts explaining how all of these things are happening in concert together. So that in the case even of a patient that has a normal air gene, there may be some sort of dysfunction related to air where those T cells leak out and get out of the thymus and go into the periphery and start causing their damage. So let's think even a little bit more forward into the future, right? Is it possible that we can take the thymus and manipulate it? And you already heard that in the last session with Audrey and Louise, that we are actually marching towards engineering a thymus. But this slide that we made was actually a slide directed at the type 1 diabetes community, right? So a big thing that's going on in the type 1 diabetes community is there's this wide recognition that you need to regenerate a large number of cells that make insulin, the beta cells of the islets. And so our collaborating lab at UCSF, Matthias Hebrock's lab, has been very good at figuring out ways to take, again, pluripotent stem cells and get them to differentiate into insulin-producing cells, right? And that's terrific, right? We need a way of putting back in the insulin-producing cells in an individual that has type 1 diabetes. But if we do that in someone that has type 1 diabetes, and we even take the cells from what we call those APS cells, from a genetically identical source and put them back into a patient, somebody with type 1 diabetes is just going to reject the cells all over again, right? They've made an autoimmune response against the beta cells. So we have to figure out what we're going to do to the immune system. So imagine a world where we could take those very same stem cells and differentiate them into a thymus, right? And then we already talked about, Audrey beautifully talked about how we could re-engineer the thymus to make air correctly in an individual with APS1. Well, the type 1 diabetes people would be interested in how do we re-engineer that thymus to make insulin better in that thymus, because more than half the people that are getting type 1 diabetes don't make insulin in their thymus correctly. Right? And so then if we took an individual where we put in both the beta cells and the thymic epithelial cells that have been re-engineered, perhaps we could reprogram the immune system so that then when we put those beta cells in, they no longer get attacked, right? And so at the end of the day, this is just an example of how type 1 diabetes and APS1 intersect and how there's not only information to learn about why type 1 diabetes, but ultimately getting to a cure could be on the same path forward. All right. So with that, I just wanted to thank all of the people that have been in my lab that have contributed to this. Of course, we want to give a particular shout out to Audrey, who you just heard from. Another thing I wanted to point out in this slide is, of course, thanking the APS 1 Foundation for supporting the work in our lab. But another thing that you'll see in this slide that's important is our lab is extremely well funded at UCSF. And part of the reason why is the NIH recognizes that studying this is a fundamental process of getting insights into how immune tolerance works. And I've also been the great beneficiary of funding from the 2 largest private foundations for type 1 diabetes research. That is the Juvenile Diabetes Research Foundation and another private foundation called the Helmsley Foundation, both here in New York City. And I know that both groups have a strong interest in further understanding APS1 because they realize that this has a connection to how type 1 diabetes happens. So with that, I'll stop and be happy to take some questions. Thank you.
We have about 10 minutes for questions.
Yeah. Sherry.
Can you talk to us about area interactions with the VNTR, IDDM2?
Well, yeah, so an obvious question would be, okay, so we know air in the mouse controls how insulin gets expressed in the thymus, right? We don't technically know that in the humans, that air controls how insulin gets expressed in the thymus. And some of this has to get into the technical challenges of how we look at expression of air and insulin in the thymus. So right now we don't have a great model system where we have a cell that makes air and makes insulin, a cell line that we can keep growing and growing. So there's no such thing from a human and there's no such thing from a mouse at this point in time. So we always have to take the thymus out of an individual and look, right? And so an obvious question nonetheless is, does air itself bind to that element that we're talking about and directly interact with it? And I have to say right now I'm pretty dubious that that's the case. It could be more complicated than we know. All we know right now is the human element is this repeat of multiple copies of the same stretch of DNA. And what actually binds there in humans, we don't understand right now. The other thing, of course, that I would point out is mice, even though air controls how mouse insulin gets made, mice don't have this repetitive element in them. Right? And so, again, even in mice, we don't understand how the insulin gene exactly gets turned on. So there's a lot to learn. At the end of the day, there's a lot to learn even in that simple biology. It's a great question.
Yeah, but don't forget, in humans, the longer the VNTR, the more protective it is.
Yep, but still.
The shorter,
Yep.
the more...
I agree.
And
but,
so a mouse has nothing.
Right. Yet air is still turning it on.
And air's turning it on.
Right. So maybe it's something else.
Well, yeah, I mean, to me that makes sense.
Yeah.
That actually makes sense.
Yeah.
And I think it's probably a multi-protein, multimeric protein complex. It's not going to be in humans as simple as just ARI interacting with the VNTR.
I agree.
I think it's going to be other players.
I completely agree.
Okay.
Adrian.
Yeah, thanks, Mark. That was lovely. Just to sort of take the parallel maybe to the ultimate extreme, Mikhail in his excellent talk this morning raised the possibility of alleles of air that are not fully penetrant for multiple presentations but might be penetrant, for example, in type 1 diabetes. So is there any plan to do targeted sequencing of air in thousands of type 1 diabetics to see if that's the case?
Yeah, so broadly the question is how deeply are we looking into air just in people that have garden variety type 1 diabetes? So a couple of things have happened, right? So I don't want to get too technically into the weeds, but one of the ways that we look at genetic variation is using SNPs, these little markers that are spread across the genome. And so there's this thing called an immunochip that we use to look at disease, and air is on the immunochip. So in fact, people are looking. The problem with the SNP on the chip, excuse my rhyming there, right, is that it looks only at the common version of air, right? It doesn't look at the rare versions of air. But now we're moving into this world where the genome is becoming cheaper and cheaper to sequence. And we are going to move to this world. And I have no doubt that we're going to start finding even more patients that are getting type 1 diabetes that have an air direct problem. Since you raised the issue, I think another important point to make here, right, this was about type 1 diabetes, but reinsert many other autoimmune diseases with this model, right? So thyroid autoimmunity, Graves' disease is another common autoimmune disease, and people have looked at the genetics of Graves' disease, and then one of the variants they get is the TSH receptor, which is this receptor that's made in the thyroid. Well, it turns out that the variant probably has nothing to do with that. It has to do with how it gets expressed in the thymus. And so we keep seeing this theme in other autoimmune diseases, right? So it even goes beyond type 1 diabetes. Oh, Einstein has a question.
Since we're talking about insulin, and insulin is secreted and in the circulation all the time,
Yeah.
how do you envision, how is the attack on the beta cells being made by anti-insulin reactive cells? I mean, is it sitting also on the surface of the cells, or?
Yeah, well, that's not really well known exactly what concentration or exact amount of insulin we need in the thymus to get rid of T cells, right? It's true that there is some insulin that's put into the secretion, gets into the blood, and probably ultimately lands in the thymus. So the simplistic way that I like to think about this right now, and we still need experiments to prove that this is true, has to do with the affinity, the strength of the T cell being able to see insulin, right? So I think that the insulin that gets expressed in the thymus, probably what it's doing is getting rid of those T cells that have high affinity for insulin, right?
But when you have the autoreactive T cells, let's say you have more in an ER deficient person out there.
Yep.
But how does the beta cells get attacked?
Well, that goes back to my super complicated slide with all the arrows and so on, right? I mean, there's many other factors that go on, and right, and let's go back to the identical twins. Why is it only 50%, right? Environment, right? Their random repertoire that they generate, right? I mean, there's many other factors, and of course, another key factor for sure are these cells that we call regulatory T cells, right? Which are the policemen that prevent attack on the islets, right? And so all those interactions are important in ultimately what happens, and I think your question really comes around to APS1. Why is it, it takes years for some of these diseases to show up, and then even in certain individuals, they don't get diagnosed until they're an adult, right? This all has to do with how the complexity of how the immune system works and how tolerance to yourself is controlled. There are many, many layers in the complicated onion, right? And they all need to come apart to get autoimmunity. All right. Yeah, thank you.
Mark, thank you so much for that talk. You answered my call brilliantly, as I knew you would. Thank you very much. That was fascinating. And as we, as a young foundation trying to organize a fairly rare disease, we've come to realize that now that we are organized as a group, we need to connect with other disease groups in order to maximize all of our potential and to cure all of our diseases. So we take that message to heart also.